Okay. All right, good. So it looks like your network is not doing very well,
It's
but
not stable.
not
Yes.
stable, yeah. But which of my screen can you see
The one displaying black color.
that has all this normal stuff, right?
Yes.
Okay. All right. So, okay. So basically, what we're going to do today, ooh, so basically what we're going to do today is let's see. Okay. Mhm. All right. So let me go through Ziegler paper for you so that I think he's kind of following this paper, right? So I'm recording this as well. After
Yes.
this, I'm going to send it to you. Let me put this right here too. So I will send it to you and Pelumi so that both of you guys can watch. So you guys have done, this is like the whole workflow of what you guys are doing. So you guys have made tremendous amount of progress. So you've done the vaccine construction. So you've done the structure refinement, right? So,
Yes.
but there's one more thing in this structure refinement, which is the disulfide engineering or
Okay.
this physical chemical parameter. You guys have done the physical chemical parameters. So, if we can't get to the disulfide engineering today,
Yes.
we will, or let me say, we should just be able to just do the disulfide engineering. Let's start with that. So the reason why you're doing disulfide engineering is because if you engineer disulfide bonds, it can help to stabilize your vaccine very well. For example, maybe sometimes you need to transport your vaccine from one place to another, just like when you spoke about long chain transport, right?
Okay. Long-term immunity.
Long-term transport.
Okay.
So if you engineer disulfide bond in between like your vaccine, when you are transporting it like long term, that can help the stability of your vaccine. That can help it to be very stable rather than like before you take it to the end point of where you're going to, it's degrade. So this is the website that you use to engineer the disulfide bond, and it's like pretty straightforward. Let me switch up my screen that you can see. Okay, so you can see. Okay, good. So to do this is very simple. You have the PDB file. And remember, from now onward, the PDB file that we'll be using is the refined PDB model that you've obtained.
Okay.
So you have these two PDB model. You said the first one is the unrefined, right?
Yes.
So this is unrefined. What's the name of your vaccine or something? Let's say unrefined structure.
Uh, of cholera vaccine.
Cholera vaccine. Okay, so we can say unrefined structure. This is refined structure. All right. So from henceforth, what we'll be using is the refined structure.
Okay.
Yeah, and that's because if like you would have seen from the Ramachandra plots as well, that the unrefined, you have a lot of amino acids that are outside their normal position.
Yes.
right? But after you refine them, you see that a lot of or most of those amino acid residue find themselves back into their like native position. So that's why
Yes.
from onset we'll be using from now on we'll be using the refined structure. So what you just have to do for this is to choose. So I just have to like go to the directory where do you know how to like navigate to directory to see where your structures are?
Yes. Yes.
Okay. So we just select refined, you click OK, then you click on upload. So once you click on upload, so there are some of these options. So these options include intra-chain. So intra-chains means you can form disulfide bonds. Okay. So I don't know if you guys have done biochemistry. Like if you guys have touched,
Biochemistry.
yeah, like if you guys have touched amino acids.
Yes.
Okay. So primarily you will know that we have 20 types of amino acid, right? 20 amino acid,
Yes.
not the types, 20 amino acid residues. We have methionine, we have glycine, we have arginine, we have
Leucine. Yes.
leucine. So these amino acid residues, they are what form the backbone of a protein structure. Because in protein structure, you have secondary protein structure, right? You have tertiary protein structure, right?
Tertiary
Right.
protein structure.
Okay. So this, what was it called? This amino acid residue, when they are joined together, you call them like a peptide sequence, right? So a long chain of peptide sequence is what now results into like a whole protein. So, but the two amino acids, no, just one amino acid that is predominantly known for forming disulfide bond is known as cysteine. Are you there? Oh, I think I lost you. Okay, let's wait for him to be back. Let me just stop recording. And Moses back.
Yes, I can hear you.
Okay, you can hear me. You're back. Okay, nice.
Yes.
Okay. So if you look at this structure now, this is the structure of cysteine. Wait, let me see.
Mhm.
Yeah.
16.
So this is the structure of cysteine. This is another cysteine. So you will see that each cysteine have like a they have a sulfur, what is called like a sulfate group at the end. If you look at this one, let me see. So you see that they have like a sulfate group, this SH, right? Then once they want to form a disulfide bond, they will, there will be lots of this hydrogen. So there will be disulfide bond between the two sulfur. So based on that, these two disulfide bonds that you're seeing, as sulfur connected to sulfur, they are very important in protein stability. So that's why you can also engineer disulfide bond in your vaccine. Because if you try to engineer this disulfide bond, it can really, really help, like maybe the stability of your vaccine. Importance of disulfide bond in proteins. So you can see that disulfide bonds, they are shown to stabilize proteins by maintaining overall structure. So they form both inter and intra bonds. So that is where this option is talking about. If it is
Uh,
intra, intra bond is within, right?
within.
But
Yes.
inter is like, let me see if I can see an example. Inter versus intra disulfide bond. Let me see, let me see, let me see. Okay, yeah, this is an example. So look at this. Can you see my screen?
Yes.
Okay, so if you look at, let me see if there's a way to make it more bigger. Okay, yeah. So if you see protein A, protein B, protein A, protein B, imagine this is one protein, right? And this
Okay.
protein B, right? Intra is disulfide bond between this same protein. But this is intra within. But inter is just disulfide bond of one protein with disulfide bond of another protein.
Okay.
And if you want to relate it to real life scenario, it's like you want to form disulfide bonds within Lee City University students. Intra-disulfide bonds is a bond between a biochemistry Lee City University student and a microbiology Lee City University student. That is intra, because both of you guys are still within Lee City University, right? But the moment you're trying to form a bond between Lee City University biochemistry students or microbiology students and University of Ibadan biochemistry students or
That
microbiology
is
students, that is inter, because both of them are not in the same, they are different, right? So that is this option. And there is this also another option for like beta, alpha beta for glycine, which I think for now you should not worry too much about it, because it's just talking about um like if you have a glycine structure, the beta carbon, the rotation of that beta carbon. But don't let me like get too complex with that. Then you also have angles. So these angles are like properties of when you have a disulfide bond. These are like properties, like the angles between, that cysteine and another cysteine, like they have to be within these angles. So they call this like a dihedral angle. So we can leave it at default, but you see that in just to have consistency in what you guys have been doing, let me see the default parameter that Ziegler used. Okay, so he used plus or minus 5. So what's choosing plus or minus 5 means is that if you choose 30, that this is what they call mean error. Mean error is if something is like plus or minus, it means it can be minus, or if you choose plus or minus 30, it means it can be higher than this number. It can be lower than this number by 30, or it can be higher than this number by 30. And what that means is that in that sense, if you're choosing like a very high error value, it means you're giving like too much space to accommodate as much things as possible, right? So this is another paper. Let me see what they used here. Disulfide engineering. Mm. Okay, are you there?
Yes, sir.
Oh, okay. Yeah, so Ziegler actually used plus or minus 5, and that's because you don't want, it's called the range of values. You don't want that range to be too much. Some people just leave it at default values, because if you leave it at default values, it means it can try as much as possible to search for all the possible disulfide bonds in your protein structure. Right. But if you like if you try to the range, if you try to lower it to something of 5, it's going to be very strict with, like, okay, we're very sure that this thing can form disulfide bonds. Then you just click on run.
Okay.
And okay. So okay, I guess I should also demonstrate that example to you. You see that when I use 5, I only have one. I will still explain this, but you'll see that when I use 5, I only have like one amino acid that can form what's it called, like a disulfide bond, right?
Yes.
But let's try and use, let's say 10. You see that now I have 3 like amino acids that can form disulfide bonds here. And I will still
Yes.
explain it later, because you'll be seeing that, oh, this is not 16 now, but I said it's only 16 that can form disulfide bonds, right? We are still going to change this to 16 later. So let's say we are now using 30, which is the default value. Then click run. Now you see we have like bunch and bunch of disulfide values. Right. So that is why it's actually good that you can test all of that. But you might actually now be wondering, so which one do we choose? Let's say we go for the default value, right? So to choose which one will be able to form disulfide bond there, we need to look for the kilocal per mole. So the kilocal per mole is like our cut off. So the cut off is, let me see if Ziegler has it. So Ziegler here, they reported that anything less than 2.2, which means that anything more than 2.2 cannot form a disulfide bond.
2.2.
Are you following me?
Yes.
Okay. So then look at this paper too. So this is another paper too. Let me see. That's bond. Okay, yeah. So this one's use 2.5 kilocal per mole. Are you following me?
Yes.
So Ziegler 2.5, 2.2. So that's still like within the same range. So I think um because I actually think the wait, I'm coming. Because I actually think they have it like in this paper where they kind of like tell you, or is he actually in this journal? So that I can just tell you, because Ziegler is using 2.2, another one is using 2.5. I'm coming, Ola.
Okay.
Hi. Oh, sorry, it's people call me and I just
I don't have a phone. I've
oh
been knocking though.
oh sorry, the door is open.
Yeah, yeah, yeah. I know, I know.
Oh,
I
okay.
was just, I like to knock first before
Oh,
you
okay. Oh, okay. Yeah, thank you very much. I appreciate that. Okay. So let me see if it's this. Is it this paper? Because they actually give like for it to form disulfide bond, there should be oh, okay. Okay. So if you click on help, I just want to show you this. Are you there? So I just want to show you this because people might be like, okay, so what is the cutoff? Just like they were asking you the last time, right? What is the cutoff?
Oh, yeah, okay.
So if you come here, I think they actually show the cutoff that should not be more than that. Okay, yeah, I think it should be somewhere around there. Yeah, so you see here that they said the bond length for a disulfide bond is between 1.81 to 2.04. So that is like the strict cutoff. But generally, you can extend it to between 2.2 to 2.5. So, so we just have to do this again. Refined. It's very simple. Then you click upload, right? So the value, let's go by, are you there? Are you there?
Yes, sir.
Okay. So since, you guys have been following Ziegler paper all this while, so let's try to keep the values the same as much as possible. As you've seen, I think the most stricter value is like 2.0, but you can still be lenient enough and still use like 2.2. Or as you can see in the other paper, they use 2.5. So let's just keep to what, Ziegler used, which is 2.2. And the value here is 5. Then you click on run. So it means that, you see, if we extend this value, because we don't want any values more than 2.2, right? So even
Okay.
though if we extend this value to 30, and you see that we are having values that are like 2.1, 2.17, 1.32, 2.52, right? So the cutoff is that we only need the values that are below 2.2. So you can also use 20 because I'm saying 2.17 here. Because if you use 5, I'm not saying that 2.17. Okay, so these are like the two residues that we can mutate to cysteine to form disulfide bonds. So let's try to lower it down to, say, 15. And let's still see if those two numbers, those two cutoff will be there. Okay, so it means that the best cutoff that we can use is 20 right here. So with 20, we can have two residue. This is called
2.17.
asparagine, 2.17 and 1.32, right? So we
Yeah,
only need
nice.
two of these. Remember that I told you it's only cysteine that can form disulfide bonds, right? And these are not cysteine.
Yes.
So what we need to do is to change this to cysteine. And what do? We mutate. That's the word. It's called mutation. Mutation, just think about it as changing something. You want to remove A, you want to change it to B. So mutation in the context of protein structure is you are removing one amino acid, you are replacing it with another amino acid. In this case, we want to replace asparagine with cysteine. So that is what mutation is called. So the process of doing that is what is
Okay.
called mutagenesis.
Mutagenesis.
Yeah, the process of doing that. Let me see, let me see, let me see. I'm trying to see if um, trying to see if there's like a picture to represent something. Yeah, this is a good example, which is instead of you having glutamate as an amino acid, you replace it with valine. And I think this is what is causing sickle cell anemia. When they say that people have sickle cell mutation. Normal protein, I'm coming, let me see. Okay, this is good because, okay, yeah, so, when they say that somebody has, is a sickle cell patient, you will actually be wondering, why does this person even have sickle cell? So it's because of mutation. So we are not sickle cell, right? We have this amino acid called glutamate. But in people that have sickle cell, that glutamate has been mutated to valine.
Really?
So you see, they put here mutant. So that's what mutation is called. One amino acid changing to another. Okay. So to do that, we have these two values. So you just select them here. You select the two, you check the boxes, then you click on create or view mutants.
Okay.
Then you click on that. A residue has been selected for. Okay. So it's actually still the same 165 as same residue. Okay. So it's saying okay, so the reason why it's saying that we are selecting, let me see the message. You see, it's saying this residue 168 has been selected for more than one disulfide. The reason is because here we have 168, right? And here
Yes.
we have that same 68. That's because the moment we
Okay.
locate it here, it's gone. We can't have it again. So with that, I would just say we go with more strict here value, where we just have just one and we just mutate that. So we just have these residues and we can just mutate those. So do you get like the concept of trying to do disulfide engineering now?
Yes.
Okay, so you create this and you have, so this is basically it. So this structure now, it has now been mutated. So now, should I supply the comments? So if you see this yellow thing here, you see this yellow,
Okay, I can.
you can see it.
Yes.
So that is actually the position of the residue that we actually mutated.
Oh,
So if you click on show disulfide bonds, if you uncheck, you see that that thing is gone. But if
yes.
you click on it, you see that it's back. So now we will now
Okay.
see the mutant structure. So I will actually
Okay.
now show you that that thing has actually changed. So it's downloading, okay. So it's done downloading. Let's go to download mutants. Okay. So let me copy this and let me go put it in your folder. Let's see desktop. Okay. Now you see that we've created a mutant, right? So we
Yes.
changed. So what we did is, that, oh, why is this thing hanging? We changed, I think the amino acid we changed is glycine, right? GLY at position 168, we changed
Yes.
to disulfide bond.
Yes.
Okay. Let me see. Yeah, let me just rerun this again. Because I want to go and open PyMo now, and I will show you that we've actually, so we mutated glycine. So GLY, do you know about you know about amino acid, right? Do you know about the 3 letters code and 1 letters code?
Okay.
Yeah, so in amino acid, we have amino acid 3 and single letter code. Okay, okay, good. So let me see. Why is this thing bringing me to research gate? I don't want that. I just want something simple. Okay, you can see my screen, right? So these are the 20 amino
Yeah.
acids. So you can write them in full, or you can just use 3 letters to denote them, or you can just use 1 letter for them. So are you getting it now?
Yes.
So if you remember, what we mutated is GLY, right? So that's 3 letters code. Are you there? Hello? Maybe you can off your video. You can just off your video. Yeah. Can you hear me now? Hello?
Yes, I can hear you.
Okay, all right. I think the video works. So,
Hello, I can hear you, sir.
okay. So you see that we mutated GLY, right?
Yes.
And also we mutated ASN. So it means that where this GLY is, we will put a 16 there. Where this ASN is, we will put a 16 there so that they will now be able to form disulfide bonds between each other. Do you get the concepts now?
Okay.
So now what is GLY and what is ASN? So if you come here, GLY is what? Glycine.
License.
So what is ASN? Asparagine. So just because like when I'm mentioning oh glycine, asparagine, I don't want you to feel like, oh, where did I see that? And if anything is not clear, just call my attention to it. Okay. Are you there?
All right. Yes, I'm with you.
Okay. So now let me open PyMo. So I'm going to load the two structures. Open immunoinformatics, refined. So let me load the mutants first. Then I will load the second one too. Open the refined one, right? Okay. So good. So we have two structures here. So we can, let me see. This one, action, color. The red. So this is the first, oh, where is it? I'm coming. Hmm. Yeah, because both of them are like exactly the same thing. So they are overlaying each other. But in PyMo, are you seeing my screen?
Yes, I can see it.
Okay. So can you see this S here that I put my cursor on? Can you see that?
No,
Okay. So let me.
no.
So can you see this thing here? It's like a movie. It's like you want to play a movie. Then there's like a forward thing there. Then there's S. Did you see that?
Okay, yeah.
So if you click on that S,
Yes, I'm finished.
it's going
Yes.
to show the sequence. So do you see?
Okay.
If you toggle it on and off, it's going to show the sequence. So this is your vaccine sequence.
Yes, I can see it.
You can see it, right? Okay, good. So this is your
Okay.
vaccine sequence
Yes.
that you actually use to predict this structure. This is what give you this structure, right? But if you could remember,
Okay.
you see that we mutated 168 and 171, right? So now let's
Uh,
168 and 171. So this is 160, 168. So this is 166. I'm coming, let me see. 168, right? 166, 167. So this is 168. Did you see? Hmm.
yes.
So this 168, that's the original structure, right? So
Okay.
in but if you go to the mutants that we created, you can see that now we have C. Did you see what I'm saying? Let me see.
Yes.
Let me show it as 6. Let me show these ones. So that's the color by yellow. Okay. Then let me follow by, let's say green. Okay. So if you look here now, you see that. Can you see my screen?
Yes, I can see it.
Okay. So originally in so this the red one is our original structure. You see that what is there is G,
Okay.
right? And that G
Yes.
means
Yes.
glycine, right?
Yes.
Okay. But if you look at the mutant, which is the green, you will see that that G has now turned to C. And you will see that the structure of this thing
Okay.
has now changed.
Yes, to yellow.
So it has now changed to yellow. So that's what they now say, that we've now been able to mutate this thing from glutamine to glycine. Are you getting the point now? So now let's
Yes.
go to the other one, which is at position 171. So if you go to 171, so this is 171, right? And how did I know? So that's why I'm saying that
Yes.
knowing the single and like this amino acid chart is very important. Did you see hen? You see that it's also the same thing as ASN.
Yes.
Okay, so that means that anywhere you see N, it's still the same thing as N. This one is just one letter. This one is just
Yes.
letter. So
Three
most
letters.
softwares like Pymo, Chimera that you'll be using, most of them use this one letter code. So that's why
Okay.
it's just good to like know, and you don't have to memorize everything. I also don't know everything. Sometimes I just like I'm showing you now, I just go to Google and just like, okay, what is this one? Sometimes I'm struggling to like remember it. So this ASN is also N. So if you come here, sorry, if you come to 171, you can see this N as well, right? That is the original one.
Yes.
So let me show this one as
Okay.
uh so where is it? Just 6. Then let me also color it as green. So now what is here is asparagine. That's the original thing. But we've now been able to mutate it to 16 sticks. Now show us what color should we give this one? We should give it yellow too, right? So
Yes. Yellow is okay.
now let me show you something. Why we are calling it disulfide engineering. You see that the red, that's our original structure, right? So let me hold the,
Yes.
that is the red. You see that when I'm showing the two, glycine and asparagine, there is no thing bonding them together. But now let me,
Yes.
do you now see the green one, our mutants? They are
I
not
can see
going
it now.
together. Hmm? In fact, let me use another
I
color.
can see it. Yes.
You can see it, Abby. So let me even use another color for it so that you can see the
Yes.
white bonds very well. Let me say magenta. Then let me show this one so as magenta. Okay. Okay. Oh, I'm thinking it's supposed to actually, okay. But it's showing that they are now connected. So it means that you've now been able to engineer disulfide bond in your structure. So now we'll save this so that in your presentation, you will show before engineering and you show after engineering. Did you get the points now? Hello?
Okay.
Yeah, so now we are now done with disulfide.
Hello. Yes, I'm with you, sir.
Yeah, this is just the network. So now we are now done with disulfide engineering. So we can move to the next one. Um, so the next one is immune simulation. Let's do immune simulation. That one too is also very fast. So to do
I think protein docking.
you said what?
I said the next one is protein docking. So,
Let me see. Oh yeah. So it's protein docking, right? And it's also simulation, right? So,
yes.
hmm, yeah, this is the right way to do it, but you necessarily don't have to do protein docking before you do immune simulation. So you can do immune simulation before
Okay.
doing protein docking. They are like independent of each other, but you have to do protein
Okay.
docking before you do molecular dynamics simulation. Are you getting the point now?
Okay.
Yeah, because this protein docking, I want us to do
Yes.
last because that one might just take a little bit of time. And what's the time right now?
This is 11 o'clock,
Yeah,
11 p.m.
okay, yeah, I want you to go to bed like 11:30 or say max 12 so that you can have like good maybe 6, 7 hours before your class. Okay, so let's do immune simulation. So can you replicate this disulfide bond on your own?
Yes, I'll try it out tomorrow. I'll try it tomorrow.
Yeah, so if you are not able to do it, just let me know and just post any question that you have. So be putting any question that you have on the group so that I can be answering it and Pelumi too can be seeing it, all right?
Okay.
Yeah, okay. So let's see. Okay, so what we want to do is immune simulation, right? So the server to do immune simulation is called, I can search for it too. If you search for it, immune simulation server. So this is the most kind of widely used server, this one. This C in sim something something. Oh, what is this? Oh no, this is not the right one. Ah, okay, yeah. So this is it. Or another way is okay, let me do it this way. Let me do it this way. So I'm going to send all of this link to do all of those analyses. I'm going to send them to you.
Okay.
Yeah. Let me see. That's so quiet. Let me send the one to do that. So file engineering. Copy. Okay. Simulation. Yeah. The new simulation. So for the immune simulation, you have to like register. I can't remember if I've registered before. Yeah, it's saying duplicate entry. That means I've registered before. So I just have to log in. But for you, you will have to like log in, right? You have to create a new account for yourself.
Okay.
Okay, so now I'm here because I just clicked login and now I'm here. What's that? Okay, so this is what the interface looks like. So basically what you just have to do is, you see here, paste either a sequence or several sequences in faster formats. Oh, okay, this is because of the previous jobs that I've done before, and I just did not terminate them. And still, are they still running? Should be a way to delete all of these things, right? So basically what you just have to do is to just copy your file as a faster file and paste them here and click on submit jobs. All right.
Okay.
So
Okay.
if you call me, let me open the file that you sent to me so that I can copy your vaccine sequence. Okay, this is your full vaccine sequence, Abby.
Yes.
And you know a faster file usually start with this, right?
Yes, I know.
Yeah, so you can just say, you can even just put a name, just say vaccine, right? Then just put Control V, just paste them there. And all of these parameters. So when we are actually now going through the results, I will explain what all of this means. But I believe you must have done all these major histocompatibility class 1, class 2.
Yes.
Okay. So this is basically to simulate, like, say, if you give these vaccines to somebody, what's their immune response to it? You know, vaccine is like a foreign something, right, that can elicit an immune response. So all of those immune response that it can actually elicit is what we're trying to simulate. So you just click on submit job. Let me see. Oh, what happened? Okay, that's the problem. Oh, just okay. So I think he does not like the way those things were cutting up and all of those things. Okay, so you just vaccine, all of these things are what we'll explain. So you just click on submit job. But why is the server not? What's happening? So normally it's just supposed to like run and just like give an output, but okay, now it's going. Oh. So it's just because it's the server, you just have to be patient with it, I guess. Because I think a lot of people use this server for immune simulation. Um, so now you can see your inputs. So these are like your input file. You see, this is what it looks like. This is your sequence.
Okay.
So if you want to look at the results, you just click on your outputs. So it's very fast. Did you see?
Okay, I can see it.
Yeah, so it's very fast. So all of these things that Ziegler have on his paper, all of these things. Did you see all of these graphs? The immune response profile. So, so that's all of all of those profiles here. Then you can click on download PDF reports.
Okay.
Gives you these PDF reports. I will download this one and send it to you, but you should also try this as well. So yeah, so that's immune simulation. I want you to try it. Then tomorrow we'll meet and I will just explain what each of the graphs mean. But at least you should try it. All right.
Okay, I will try it.
Yeah. So that's for that. And you can adjust, the parameters and all of that, but those are things we'll discuss tomorrow. Let me see Ziegler, if he used if he adjust any parameters or anything like that. Yeah, so next we'll be going to protein docking, immune simulation, the time step of the simulation receptor, why the simulation step was.
And we haven't done this thing, population coverage.
Oh, really? I thought you guys have done population coverage.
No.
Oh, okay. Okay. Hmm. Okay. Hmm. Uh, so he modify like the time steps, he changed the time steps as well. So that's something we can also like look at why they okay. Hmm. So the next thing that I will do is we said it population analysis coverage. Oh, actually thought you guys have done. Let me see. Yeah, population analysis coverage is supposed to come before you guys do a vaccine construction.
We haven't done it though.
Oh, yeah, let me see. I think basically what you just have to do is, you just choose the word and area, country, ethnicity. Then I think basically we'll just have to like paste your epitopes here and submit the jobs. So, should we do this or should we just move to, or should we just move to protein docking? Yeah, I would say let's do protein docking first. Then once we are done with protein docking, if we can't cover it today, we'll just do it tomorrow.
Okay.
Yeah. Yeah, but you guys are supposed to have actually done that way for, and I think these people also did population coverage. Let me see. Disulfide. Mm. Yeah, I took marking cross. Well, I don't think these people really, but maybe on your card you did. Okay, yeah, on your card we did population coverage. Mm. Okay. But you still have like each of the epitopes that you predicted, right? Like the final set of
Yes.
epitopes. Yeah, so that's
Yes.
basically like what we use for. So how many epitopes did you guys predicted?
Um, Let me check. I think the total one I use for the vaccine candidates is 14. It's 14 or 13.
But which one did you use for like to construct the vaccine? How many of them did you select to construct the vaccine?
And that's what I'm saying. 1, 2, 3.
Oh, you used 14, right?
13. 13.
13. Okay, so you can just set this to 13. and you can select like the word, as well as, let me see. I think you should be able to like select West Africa, particularly like Nigeria, because that's like, yeah, so select West Africa. Let me see. Like Nigeria. So yeah, I would say you should send like the list of all of those epitopes to me so that I can
Okay.
submit it. But as what was it called? You have seen as well, we can easily just paste the epitopes here. Then the MHC restricted alleles. Did you guys, during like you guys initial analysis, did you get like some restricted MHC?
Yeah, I don't think so.
Okay, if you did not, but just send me like the list of all of those epitopes so that I can actually do that sound.
Okay.
Okay, yeah. So what's next? What do we do? What do we do? So we want to do protein docking, right?
Yes.
Okay, good. So for the protein docking, the reason why we are docking is to know like the binding affinity between your vaccine and the target receptor. So in this case, you are using an adjuvant that targets, is it toll-like receptor 2 or 4?
Um, so like receptor 4 agonist.
Agonist, okay. So Ziegler was saying that one of the proteins that you chose, obuin, that it stimulates immune response by targeting toll-like receptor 4. So we are going to use both of them. So we are going to use both toll-like receptor 4 and toll-like receptor 2. All right.
Okay.
So now, we have your home protein structure, the refined one.
Yes.
So let me see. So we'll have to download the receptor. And to get that receptor, we'll use Protein Data Bank. Have you heard of Protein Data Bank before?
Yes.
Okay, good. So Protein Data Bank is like a public database that houses protein structures where they are being deposited. So you just search for Protein Data Bank. So there is a search button right here. There's a search button. Why is it so big like that? Okay. So this search bar here, you can actually search for toll-like receptor 4. Toll-like receptor 4. So you search for it, and it's just going to load for a while. And we just wait for toll-like receptor 4. This is toll-like receptor 1. Yeah, these are not toll-like receptor. This is toll-like receptor 7. So you can restrict your findings to like Homo sapiens so that it's humans, or you can say toll-like receptor for I think Ziegler actually has one. He had one in his paper. Yeah, the toll-like receptor 4 that he used. So each protein structure usually have their own PDB ID. So those PDB IDs are 4 alphanumerical numbers. So if you copy this PDB ID and you paste it on PDB, so you can see entry search terms, entry ID, or sequence. So if you paste
Okay.
it here, you can see the protein, which is
Oh,
the structure of human CLR4.
okay.
Or you can also search like CLR4. I think I was using the wrong, what's it called? You can search TLR4 in complex with an inhibitor. Yeah. So this is TLR4. This is one TLR4. This is another. So usually there are like multiple protein structures that are deposited in protein data bank. You just have to like look for which one do you actually want to select. And there are selection criteria for this. So if we want to select the one that, is from human, you can just come here and ensure that any of the one that will be shown here should be human. Let me see, eukaryote, X-ray. Okay, so you can see TLR4 in complex, like lots of TLR4 here, right?
Yes.
Yeah, but let's go with the one that Ziegler used so that we can just use that one. But I mean, that does not mean that we can actually like, select a new one. I think, is this the one that I used? So this is the one that I used, right?
Yes.
488A. Yeah, so that's the one that I used. So I didn't mean that, Ziegler has not done this before. We'll actually, just choose, we'll carefully select one to use. But since he has used this, let's use this as well. So this just tells you like, oh, crystal structure of TLR4, blah, blah, blah. And what is most important is this. One of the things that is also important is this global stoichiometry. You see that it tells you heteroforma. What that heteroforma means is that it's 4 chains. But from the word hetero, it means they are different. So it means they are like different chains. But if it is homoforma, then it means like, oh, okay, they're actually the same thing. So you just have to scroll down. Then to see your toll-like receptor, it is telling you like your toll-like receptor 4 is either chain A or chain B. So, sorry. And chain C or chain D is this lymphocyte antigen. So now we'll actually download everything. Are you there now? Hello?
Yes, sir. Yes. Yes.
So you go to download files, you go to PDB format, you can just click on it and it will download. Did you see now? It has downloaded. Or let me see if this works. I just usually just click save link as so that I can just go to that a directory where I just want to save. So now the next thing is, you know that I just told you that it's not just one chain, right?
Yes.
So let me delete this. Action, delete object, action, delete subject, action. So there is nothing there. So now I will load in the new structure that I downloaded. Did you see the new structure now? So now I can
Okay.
color them by chains. You can come to this C. You will see color by chain. Did you see? Then You can color by chain. So the one that the chains are the same, you see that this chain, and this one, they are actually the same thing. I think you can actually split chains.
I can't see your screen again. Oh, I think it's my network also.
Oh, I think it's your network.
Okay, okay, I can see it now.
Okay, you can see it now. Okay. Mhm. Okay, so these are different chains. I think you can actually come here to show chains. Um, wait, edit, display sequence mode, chain identifiers. So if you go to chain identifiers, you can see that we have chain A, right? So that's chain A. Did you see where I just went to?
Yes.
Display sequence mode,
Yes.
chain identifier. Display sequence mode, I can change
Okay.
it to residue's name. So
Okay, okay, I can see.
yeah, chain identifier. If I select chain A, you see that that's everything that is chain A. If I select chain B,
Okay.
that's chain. So your toll-like receptor is what? Uh, where is PDB? Yeah, so you see that your toll-like receptor is what? Chain A or chain B. So that means the only one, and we can only use one of them because they are both the same thing, right?
Okay.
So, mm, so this is chain A, this is chain B, this is chain C. You see chain C there, right? Chain D, we don't need those.
Yes.
Chain E, chain F, G, H. So we don't need all of those ones. So you can click on them and you can, so if you select them, you just right click on any one of them, then you can click on remove and they will be gone.
Oh, okay.
Yeah. Then also you can click on this action, then you click on remove waters. And the reason why is because we don't actually want waters during molecular docking. So you click on remove water. You see that all those red dots, small things now, they are gone.
Yes.
Okay. So we've removed waters. So now we only have just two chains. So what is this? Okay, so this is part of. So now both of these are actually the same thing because we can extract these. I want to show you that they are actually the same thing. Action, extract objects, action, extract objects. So now you see that we have chain A and chain B. So we can turn on the sequence and show sequence mode, sequence name. So both of chain A and chain B, that's what I'm showing, right? Oh, okay, what's this? This is like a nag. Okay, so we don't need this to remove this. We don't need this as well. Oh, that's a nag as well. So we remove that. So all the ones that I'm just removing, they are like what they use in solving the structure. So we actually don't need them. So the only thing we need is just the structure. So now,
Okay.
just to show you that both of them are the same, if you click on chain A, where is chain B? So if you click on chain A, you can see A and you can see B, right? So you see that they are
Yes.
actually supposed to be the same thing. Let me see. Prevenine 16. Yeah, okay. Yeah, they are actually the same thing. Uh, what's happening? Isoleucine. It's looking like there is a shift. Valine, valine, glutamine, glutamine. Okay, so what you can do is you can actually align them. Align to so you can come here, align to molecule 4G. Now you see that both of I hope I'm not going too much.
No.
Okay. So what I just did is I was trying to be sure that they are actually the same thing. So that's why I aligned them. So this is called alignment. And you can see that both of them are actually like perfectly the same thing. So if they are perfectly the same thing, we only need one of them. All right.
Okay.
So, okay. So all of them are the same thing, starting from this residue here, right here. So you see that we have tyrosine, tyrosine, glycine, glycine. So these ones, they are
Okay.
different because there is what we call um, hmm. So these ones, they are different in the structure because they are like maybe just one part that they need to attach to the structure to use that to solve the sequence. So, but apart from that, you can see that everything is the same, right? So that
Yes.
we just need one of it. So do you understand why we just need just one of it now? So this one we can just delete it. Uh, this one we can just delete it. So that way we just have just one. All right. So now we'll just have to save it. Go to File, Export Molecule. All right. Then we'll go to Enabled PDB option. So you just save. And you see these drop down options here, right? So you just click on PDB,
Okay.
then you give it a name. So let's give it CLR4.pdb. Then we click on save. So now we are done with TLR4. But let me explain to you the concepts of docking. So in docking is basically you have a ligand, all right? And you have a protein, which in this case is your receptor, right? You see, it's like a lock. They call it
Okay.
a lock and key hypothesis. It's like you have a padlock and you have a key, and you just want to put your key into the padlock, right? Just to see how well it's fitting.
Okay.
Then if it fits, then you know you can open that padlock with your key, right? But if it does not,
Yes.
even though it's enter and it does not fit, you can't open it. So that's the concept of molecular docking. You want to know your vaccine, which in this case is going to be our ligand. You want to know how well can it bind to your receptor. All right.
Okay.
So to do that, we have multiple servers. We have HDoc server. So what we want to do is protein docking because your vaccine is a protein, right? The TLR4 too is a protein.
Yes.
So what we want to do is called protein-protein docking. So we have multiple servers that you can use to do this. You can use HDoc server or you can use ClosePro. So let's start with this one and submit, then we'll go to ClosePro too. So he said, input your receptor. So what is our receptor in this case?
CLR4.
Exactly. So we now go TLR4. You see that I'm picking the TLR4 that we saved out of what was it called? PyMo, right?
Okay.
So you click on open. So that one is there. So what is our ligand? Hello?
Hello, yes, sir.
So what is our ligand?
Ligand and
So our ligand in this case is going to be your vaccine. It's a protein too,
okay.
right? But we are just considering it as a
Yes.
ligand.
Okay.
So that's why you now choose your refined structure. Not the mutant one, no, just the refined one.
Refined.
So you
Okay.
click on open. Then all of these advanced options, you leave them defaults. Then you just click on submit. You see that the hardest, what was it called? The hardest part is trying to prepare the structure in
The
Python.
structure. Yes.
Yeah. So just in case you want to search for it. Okay. So this one is running, or we'll leave it like that. Then we'll go to ClosePro. So this ClosePro, I'm not sure if you can use it. It's just the same thing as HDoc, but the reason why I'm not sure you can use it is because if you are using it for the first time, it tells you to register.
Oh,
Yeah, and you need a school email address. You can't use your permanent email, your normal email address.
I think I should ask about the email tomorrow, sir.
Yeah, if you can find out, but if not, I can still like submit it for you and I even remember my username for this thing. Can't remember my. Let's just hope it's the right one. Oh, yeah. That's just refreshing. Hmm. Oh, can't remember my username. Reset password. Okay. Oh, I'm trying to reset the password. Oh, wow. This password is very funky. How do you expect me to remember this? So they sent me my password. Let's see if that works. Oh, what? What's the password they sent me? Oh, now I forgot my username too. Oh, wow. I opened this very long time ago. I know you need to go to bed now. We are almost done, all right? So log in. Let me save it. Okay, so it's just basically the same thing that we did like this server, all right?
Okay.
So what we'll just do is upload receptor. So what is our receptor?
Toll-like receptor 4.
So you just choose PAL, click on toll-like receptor 4. So what is our ligand?
The vaccine sequence.
Exactly. So just click on the structure of the vaccine sequence, the refined
The
structure,
refined structure.
right?
Yeah.
Then, just like HardDoc, you have all these advanced options, but just leave them default. Then you just click on doc. Then let's give it a
Okay.
job name. Let's say CH vaccine, right? Vac. The reason why I'm giving it this acronym is because, it's your job name is going to be publicly available. So you don't want people to
Okay.
know what you are working on. So that's why some
Okay.
people just use like acronym that you are the only one that you know the meaning. So this CH means cholera, this VAC means vaccine. So you
Vaccine.
click on
Okay.
doc, then it submits as well. Yeah. Did you see what I was saying that people use very weird name? What's the meaning of
No.
it? So people just use very weird name. Did you see that meaning? I don't even know what is the meaning of all these things.
Okay.
Yeah. So now you see that it says Steve Balogun, right?
Yes.
Then it says that my job is what? It's now processing.
Process.
Yeah, in fact, I even need to change my username because you see that people just use username that does not make sense. So yeah, so it says that it's processing. So we just have to wait for it to be done. And this is our job ID. Can click on it. So it's processing it. You see, this is your vaccine, right? This is the
Yes.
receptor. You see that it's processing it. So we need this ID too. Just like that one of plus row job ID. Whatever. Okay, so we are done with molecular docking. That's basically it. We just have to wait for the result to be done. Then after that, we just take it to Pymo and we'll do very beautiful images for you guys. Oh my God, I wish I've done this like 2 weeks ago so that you guys can have enough time to look at this. Oh, sure. So once that is done, the last thing that we will do, you see that you have enough results to go and present already.
Yes.
So the last thing is we are not doing it now because we need to wait for that protein-protein docking to be done. Is this molecular dynamic simulation? And it's very simple. Let me show you. So you have to come to this website. So I'm sending you the links to let me send you the links to Plus Pro Protein Docking. Let me get the website. Let me get the website for yeah, so this is Close Pro. You just log in. This is H-doc too. Let me copy H-doc for you. Oh, it's gone. Hmm. So I see that Olayemi is gone. So basically, to do the I mode, what we just need is just the results from protein docking. We take it, we put it in there, then we submit. And like in 2 minutes, we have our results. So yeah, that's it. Okay. So I'll resume recording. So basically, I said the molecular dynamic simulation here. So to do the simulation, the results that we get from the protein docking.
Okay.
So you just come here, it's going to give you a PDB file, like a complex. You see the results, just
Oh,
results, then you just browse and you just click submit, and that's it. So just like the immune simulation, it will give you all of these graphs.
oh, okay.
So it will give you all of this data.
Okay.
Yeah, and that's basically it. Then this one too, we can also do this one as well. So yeah, that's basically it. So that's basically everything. At least that you need to go and present so that it doesn't get too much. I mean, this is a lot. I wish I've actually showed you all of this. Wow. I really, really wish. So what do you think? I've sent you the link to all of those things. So I just replied back to the email you sent to me. So do you think you can give this like a try or something?
Yes, I will try it tomorrow when I get to school.
Yeah, and don't let it take too much of your time. We don't need to put in all of the results. Like you see, there are a lot of graphs and you only have 10 minutes to present, right?
Yes.
So
It's
anyway,
not because when we are presenting, like the time and everything.
is what?
I said they did not, they haven't tell us the time we are presenting.
Yeah, I'm not even sure if those guys are not even going to postpone it again. But let's let's see. I will ask some of my friends that I know that are also going to the conference.
Okay.
Yeah, I'll ask them and see if they've heard anything from them. I'll remind you. I'll just let you know what I hear from them.
Okay.
Yeah, so I'm going to show it. I'm going to convert this and I'll upload it to my YouTube, like a private YouTube. Then I'll send you the link so that you can be able to watch it.
Okay.
Yeah,
All right.
okay. All right, so you should go to bed now. We are almost at 12. Bele?
Yes, sir.
Okay, let me stop recording.